Hi and good day. This is Lecture 5, Habitat Analysis and Wildlife Survey or Assessment Techniques. Wildlife species diversity and composition is correlated to habitat quality. It has been shown that intact forests, or those with low degrees of human disturbances, contain higher number of endemic species than disturbed habitats. We have already discussed the patterns of diversity for herpetofauna, birds, and mammals, and overall, intact or less disturbed habitats contain not only higher numbers of endemic species, invasive alien species are also unable to establish in these habitats. Habitat sampling techniques vary depending on land types, but for forests, on an ecosystem level assessment, this can be done by identifying the forest formation. And for the species level assessment, nested quadrat technique may be conducted for the under canopy, understory, and ground cover. For the survey of terrestrial vertebrates, we also identify the biogeographic zone, or more specifically, the Pleistocene aggregate island complex where our sampling area belongs. Identify also the elevation and predominant forest formation. Some areas have unique forest types, like the pygmy forest of Mount Hamigitan. Dr. Fernando of the College of Forestry and Natural Resources of UPLB has identified 12 types of forest formations in the Philippines. These are the tropical lowland evergreen rainforest, tropical lower montane rainforest, tropical upper montane rainforest, or what we call mossy forest, tropical subalpine forest, forest over limestone, forest over ultramafic rocks, beech forest, mangrove forest, pit swamp forest, freshwater swamp forest, tropical semi-evergreen rainforest, and tropical moist deciduous forest. How many of these have you ventured in? Within a forest ecosystem, especially in tropical lowland evergreen forests, there is vertical stratification consisting of the leaf litter and subterranean layer, which is a habitat for the fossorial or burrowing wildlife, the forest floor for the ground-dwelling species. The understory is the undergrowth or flora beneath the canopy, utilized by terrestrial as well as arboreal species. The forest canopy refers to the upper layer or habitat zone, formed by mature tree crowns and including other biological organisms. For a single tree canopy is used to refer to the extent of the outer layer of leaves of an individual tree. Hornbills are examples of canopy species. Above the canopy is the emergent layer. When do we conduct our biodiversity assessment? Let's consider the most important climatic element. For wildlife, we are interested in the 4 climate types based on rainfall distribution. We have types 1, 2, 3, and 4. Type 1 has 2 pronounced seasons, dry from November to April and wet the rest of the year. Type 2 has no dry season and a very pronounced rainfall from November to January. In type 3, the seasons are not very pronounced and it is relatively dry in November to April and wet the rest of the year. In type 4, rainfall is evenly distributed throughout the year. Knowing the climate type in the area of research is important so you can schedule when your wet and dry season samplings will be done. Wildlife species also vary in terms of activity patterns, and this is referred to as their temporal distribution. So there are diurnal species, those that are active during the day, like most birds and lizards, and the cobra Naja samarensis. Nocturnal species are active during the night, so these are most mammals and frogs, and snakes, for example, Lycodon olicus. And there are also those that are active during the twilight, like during dawn or dusk, and these are the crepuscular species. So for example, the night heron Nycticorax caledonicus, and this one, Eurostopodus macrotis, the night jar. So they are mainly crepuscular but may also be nocturnal. How will you know if your sampling is adequate? A simple method is to make a species effort curve. On the left is a graph that shows an increasing trend even after 360 net nights, or 60 nets set for 6 nights. This suggests that additional species may still be recorded. The graph on the right, on the other hand, shows a flattening of the curve at only 60 net nights until 180 net nights, or 30 nets set for 6 nights. This suggests that netting has been adequate. Wildlife assessments are done for a number of reasons, but the primary purpose is to have a baseline data. After an assessment has been done, subsequent field surveys, especially in protected areas, would be for monitoring purposes. Wildlife species may be recorded through direct encounters, usually through transect methods using visual and auditory cues, and through capture methods. Indirectly through indices of presence or interviews and use of secondary data. Published data, whether old or recent, offer invaluable data from which current data may be compared. They are also good sources of wildlife photos from which you can compare your own captures and may help in identifying specimens, especially when field guides are not available. I find these publications especially helpful for new herpetofauna species. For getting primary data on herpetofauna, the modified strip transect sampling is conducted by designating a 250-meter strip transect in suitable habitats. This is purposive, so choose areas where amphibians and reptiles will most likely be encountered. Lay out 10 alternating plots measuring 10 by 10 meters on each side every 25 meters and 5 meters perpendicular to the transect. Mark your transect with flagging tapes. The total area will be 1,000 square meters. Traverse the transect by scanning through and making searches. Your time of transect will depend on your target group. Follow auditory cues of frogs and use your bare hands to capture individuals. Record species, number of individuals, and type of encounter. Note descriptions of their microhabitats and ecological or behavioral observations if there are any, and take pictures. Your sampling will be from 7 a.m. to 9 a.m., 11 a.m. to 1 p.m., and 7 p.m. to 9 p.m. This will include diurnal, nocturnal, and crepuscular species. As with the other wildlife groups that you will survey, make sure you record important data, such as specific locality and coordinates of your sampling sites, weather conditions at the time of sampling, dates, time and end of sampling, if applicable, vegetation or habitat information. List the species you record and note other information as you deem necessary. Make sure to organize your raw data so that it becomes easily understandable even to other members of your team. Pitfall traps and glue traps are used to capture terrestrial amphibians, small lizards, and sometimes even snakes, and even small mammals. Lay out pitfall traps and glue traps. Bury plastic containers with brim lower or about the same level as the ground. Space the traps 5 meters apart. Cover the trap with natural roofing material. You may add drift fences between traps. This will facilitate movement of the animals into the traps. Alternate traps every 5 meters on each side. Pitfall traps may also be set without drift fences. This is a drift fence using tarp and with pitfall traps using soda bottles. This one is using plastic pail and wire screen. Check the traps for captures. Record amphibians or reptiles observed or caught outside of the transect sampling area. That is, during net watching, checking of mist nets, and rebaiting of traps, but within the sampling site. Collect by hand or using a handling equipment. Processing of the samples may be done to identify the species. Record detailed descriptive and quantitative morphometrics in meters and in grams. Take the biometrics, again, if this is necessary for the species identification. It is important to take digital photographs in color. If possible, use macro lens to vividly show color differences and details of morphological characters. Photograph also, if possible, their microhabitats. Properly label photographs and bioacoustic data. Release all captured and identified individuals at the site of capture. For the indices of presence, you may observe, for example, for leathery eggs of snakes, calcareous eggs of geckos, burrows and nests, snake skins, and skeletal remains, although they may not be easy to identify. Species recorded will add to the species list and provide invaluable ecological and behavioral notes. Record the GPS locations. These are examples of indices of presence. For example, the shed snakeskin, tree frog foam nest, and crocodile teeth necklace. Of course, the species identification will be difficult. Ethnobiological interviews may be conducted among local residents or indigenous peoples. Show pictorial guides for verification. Note other important information such as local names, personal observations of feeding habits, habitat use and breeding information, and knowledge on wildlife utilization. Include only those with verifiable descriptive information or other additional evidences. Note the names of the persons interviewed, date and time of interview, and GPS coordinates of the locality. Again, if you are going to interview indigenous peoples, you will need to get a permit. As with other wildlife groups, you should come up with a checklist of species that you have recorded based on the various methods you have used. Then, from references such as field guides and websites, search for the common names, residency status, habitat association, and conservation status of the species. Once you have this checklist, you can make a pie chart of the residency status habitat association, and conservation status of the species, and get an idea of the species composition in your sampling sites. For primary data on the avifauna of your sampling site, the line transect method is commonly used. Establish a 2-kilometer transect route and traverse this route early in the morning and late in the afternoon. Record the bird species and the number of individuals seen or heard. Your travel period may be approximately 2 hours per 2-kilometer transect. Use binoculars and field guides to aid in species identification. If available, use a digital SLR camera with telephoto lens and digital audio recorder. Species not validated on site can be confirmed through these websites. Netting is used to supplement the line transect method. 12-meter mist nets are set along possible flyways and near feeding trees. Nets are set individually or in a series near the ground, or a pulley system is used to reach the higher forest strata. Try to avoid unnecessary clearing of vegetation in the area, although trimming of branches of trees reaching the nets may be done. Nets are tended for 24 hours for at least 3 days and 2 nights, but this will vary depending again on your adequacy of sampling. Check periodically for possible netted individuals and ideally every hour. Carefully retrieve birds and place them individually in cloth bags. Hang the cloth bags in cool, shaded area prior to processing. If necessary for identification, take the measurements of your captured birds. Photograph representatives of species captured and then release at site of capture. Indices of presence for birds include feathers, skeletal remains, eggs and eggshells, and nests. But again, the species will be difficult to identify. Again, record the locality and GPS coordinates. Species will add to the species list but not to the computational statistical data. Also conduct ethnobiological interview in the same manner as in the herpetofauna. The survey methods for mammals take into consideration their size, activity patterns, and feeding habits. Mist nets are used to capture bats, in particular, fruit bats. These nets are set late in the afternoon along possible flyways or near feeding trees. And no, there is no need to put baits like hanging fruits onto these nets. Fruit bats will naturally entangle as they fly through the nets. For microbats, remember that these small bats can echolocate and they have the ability to detect and evade the mist nets. A few, however, do get captured, but you have to conduct net watching. So before dusk, because microbats are crepuscular, observers watch for any microbats that get entangled. They have to be removed as soon as possible because they will chew the net. Remember, they have fine, sharp teeth to eat insects. and then they can escape, leaving your net with a large hole. After net watching, or when it gets dark, you will observe that captures will then be consisting mostly of fruit bats. Fruit bats have to be removed immediately also. This prevents bats from being too entangled, nets from being chewed upon, and potential predators are warded off. Captured bats are placed individually in cloth bags, which are hanged as you continue to remove other bats. Checking of nets can be done every hour until around 10 p.m. You may leave the nets open, but make sure to return in the early morning to remove captured bats again. For microchiropterans, other methods that may be used are tunnel trap, harp trap, and the use of bat detector. Captured specimens are identified based on Engel and Heaney 1992, which is the key to the bats of the Philippines. You record the species and the number of individuals captured. Record the weight, sex, and approximate age on your data sheets. Take the biometrics, but only if you need it for identification. Prior to release, take photos of the captured bats, making sure to show the distinguishing characteristics, and then release the bats at the site of capture. For small non-volant mammals such as rats, mice, and shrews, trapping is done. I guess you might be familiar with the snap trap on the left and cage trap on the right. Your family might have used this to capture rats and mice in your house. In the field, these are also used. However, snap traps are kill traps and are not recommended, especially to capture endemic rats. Fabricated cage traps allow the capture and release of mammals. However, trappability may be low and they are quite bulky to bring in the field when compared with snap traps. These traps may be baited with roasted coconut meat coated with peanut butter or live earthworms to capture vermivorous rats. Setting of traps is done in the late afternoon. Traps may be set on the ground, positioned 5 to 10 meters apart under root tangles, in front of burrow entrances, along runways, and beside or on top of fallen logs. Traps may also be set above ground. Traps baited with roasted coconut meat coated with peanut butter are tied onto horizontal branches of trees, which may also serve as runway or walkway of arboreal mammals. To easily locate the traps, markers are tied above or near where the traps were placed. In areas of dense vegetation, it is easy to lose track of your traps. Captures are retrieved early in the morning and baits replaced in the late afternoon. Traps are maintained for at least 3 days or depending on your species effort curve. When processing the specimens, take note of the following: the species and the number of individuals caught. Note the weight, sex, and approximate age, whether they are adult, subadult, or juvenile, and when applicable, the reproductive condition. Take the biometrics, and again, only when it is needed for identification or when your study needs this biometrics. Just as with the bats, take a close-up photo of representatives of the species captured. release them at the site of capture. Camera traps may be used to record cryptic and elusive species. It is also preferred because it is non-invasive. However, camera traps are expensive and they may be stolen in the field. Here is a sample of a photo of a squirrel in Mindanao taken by a camera trap. It is not easy to get this kind of photo. Moreover, it may be difficult to identify mammals through photos only. Medium to large mammals, aside from camera trapping, may also be sampled through spotlighting, ethnobiological interviews, and indices of presence. A shows a civet eye shine during spotlighting, and B, civet scat, used as index of presence. There is also a photo of a cloud rat skull, which may be used as index of their presence in an area. Please be reminded that any method used that involves capture, whether using bare hands or equipments such as nets and traps, will require you to get a gratuitous permit. This is according to RA 9147 or the Philippine Wildlife Act. Invasive methods, no matter how careful we are, cause undue stress to animals. Not only that, this human-wildlife contact also provides an opportunity for viruses to jump to humans, and we all know the global consequences of such spillover. The paper of Zemanova entitled Towards More Compassionate Wildlife Research Through the 3 Rs Principles, Moving from Invasive to Non-Invasive Methods, discusses how we can replace, reduce, and refine our current methods. Read more of this as an attachment along with your learning activity. So this is the last slide. You may now proceed with your learning activity, and have a good day.